The 3M Allergen Protein Rapid Kit contains the following components: 26 extraction tubes to prepare extracted samples, 25 individually packaged lateral flow devices specific for each allergen, 50 mL of 3M extraction buffer, which is specific for each allergen and kit lot. The 3M Allergen Protein Rapid Kit is intended for screening for the presence of allergen proteins in clean-in-place CIP final rinse water, environmental swab samples, food ingredients, and processed food products. Before running a test, be sure to read the Product Instructions for Use document thoroughly. To analyze liquid samples, first label one microcentrifuge tube for each sample. Then measure 900 microliters of 3M extraction buffer into the labeled microcentrifuge tube. Next, add 100 microliters of a well-mixed sample. Shake vigorously or vortex for 15 seconds to mix thoroughly. Then remove one 3M allergen protein lateral flow device from the package and place it on a clean, dry, flat surface. Each cassette is labeled with the specific analyte for easy identification. Label the cassette with the appropriate sample ID. Transfer 100 microliters of the extracted sample using a clean transfer pipette or pipette tip. Apply it to the sample well on the 3M allergen protein lateral flow device and start the timer for 11 minutes. For additional questions, please consult the instructions for use available online or contact your 3M Food Safety sales representative.